Assalamualaikum and hi everyone. Today, our group will present about the quantification and characterization of airborne microplastics in the coastal area of Terengganu, Malaysia, according to the 7 steps of analysis. The first step is define the problem. What is the problem that needs to be found, and is it qualitative or quantitative or both? Based on the article, we found out that microplastics or MPs are the problem since it poses a threat to ecosystems due to their capacity to bind with toxic chemicals. It is both qualitative and quantitative because MPs are normally counted and identified using a stereo microscope based on their color and shape. Next, what will the information be used for and who will use it? From the article, we found out that the information about MPs, like the fact that airborne MPs can act as a vector for coexisting air pollutants as they travel through the atmosphere, can alarm us on the associated health concerns. These informations will be used by the Microplastic Research Interest Group, MRIG, and the Atmospheric Air Quality and Climate Change Research Interest Group. Next question is, when will it be needed? This study can be expanded to investigate the potential impacts of MPs on human health and how to alleviate their adverse effects. How accurate and precise does the problem have to be? Since the problem known is microplastics or MPs, the physiochemical properties of MPs are their small size, which is less than 5 mm, has wide variation of shapes, colors and polymers, strong hydrophobicity, large surface area and long lifetime. These information needs to be accurate and precise because it tells us that they are an exceptional pollutant in the environment and this will all affect their toxicity. To control MP pollution, we need to depend on the characteristics of MPs. What is the budget to analyze this problem? From the article, we know that it requires a low cost because a simple method is used. Lastly, is there any analyst consultation with the client to plan a useful and efficient analysis? From the article, we found out that to plan an efficient analysis, they have consulted with the client about using the passive sampling method described by Beamer et al. 2014 and Welsh et al. 2017, which involves glass petri dishes with glass microfiber filters to collect and analyze air samples, ensuring minimal contamination and allowing direct visualization of suspected microplastics.
Hi, my name is Nur Shamim binti Muhammad Azahari. My matrix number is A202906. The second one is select a method. Okay. The sample type is airborne MP samples. The size of sample characterized by their small size, which is less than 5 millimeter. And the other one is ATR FTIR technique, which is for the particle size as small as 6 micrometer. Okay, for sample preparation needed, the morphology structure of MP observed by using a scanning electron microscope to show a cracked surface on MP, suggesting weathering and irregular fragmentation. Okay, the other one is elemental analysis by using energy dispersive X-ray spectroscopy (EDS) to show the presence of heavy metal. Okay, and then the other ones MP were manually count and identified by using a stereomicroscopic based on their color and shape. For the tools or instrument available is glass petri dish that contain a glass microfiber filter, and the other one is pre-cleaned glass petri dish using stainless steel tweezers and covered with aluminum foil for further laboratory analysis. So about the cost, it is low cost because this is a simple method and easy to handle. Okay, for the speed, we have two types, which is passive sampling and active sampling. The last one is this method is not automated.
Assalamualaikum warahmatullahi wabarakatuh. My name is Ainul Hasbi binti Ajri Masidi. My number metric is 8203713. So I will talk about obtain a representative sample. The sample type is atmospheric deposition sample. Sample homogeneity is air sampling took place during a hot and dry day with moderate wind in which triplicate samples were collected over a 24-hour sampling period. The sample size is sizes as small as 6 micrometer. For sampling statistics or errors, this is the diagram for sampling statistics. So from this diagram, it shown that the most biggest is transparent white, that is 38%, followed by blue 25%, black 20%, red 13%, green 2%, and the last one is orange and purple 1%.
Hi, my name is Noor Atira. I'm going to talk about for the next step, which is prepare the sample for analysis. For the first question, solid, liquid or gas? From the article we found that it was a solid. Let's dissolve. It is a solid particle and can analyze in their solid form, so no need to dissolve. The next question is ash or digest. From the article we found that it was a digest, so filter paper used for sample collection were thoroughly rinsed with the eye water to remove impurities. Chemical separation or masking of interference needed. So from the article we found that is masking of interference. Filter papers used for sample collection were thoroughly rinsed with the eye water to remove impurities. Need to concentrate the analyte. So from the article, it's no need due to the particle in a solid form. Need to change derivative the analyte for detection. The answer is no need. Need to adjust solution condition pH at reagents. The answer is no need.
Assalamualaikum warahmatullahi wabarakatuh. And my name is Salmi Anis Mulia binti Abdul Aziz. And here I want to talk about question 5, where we should perform any necessary chemical separation. And there is 6 chemical separation technique. The first chemical separation technique is distillation, where distillation is not directly applicable to the analysis of MPS, as it is typically used to separate components of a liquid mixture based on boiling points. However, it could be useful in preparation of solvent or in purifying reagent used in other analytical techniques. And the second technique is precipitation, where precipitation could be used in the preparation of sample to remove interfering substance from a solution before further analysis. For example, precipitating dissolved organic matter that may interfere with MP identification. The third chemical separation technique is solvent extraction, where the purpose of this technique is to separate compounds based on their solubility in different immiscible liquids. Their procedure is: first, add the mixture to a separatory funnel and add the extracting solvent and shake the funnel, allowing the compound to partition between the two layers. And then let the layers separate. Drain the layer containing the desired compound, and the last one repeat if necessary for further purification. Next is solid phase extraction (SPE), where the purpose is separate compound based on their affinity for a solid adsorbent. The procedure: first, condition the SPE cartridge with a solvent, and then load the sample onto a cartridge, then wash the cartridge to remove impurities, elute the target compound with a suitable solvent, and then collect and concentrate the eluent if needed. For next chemical separation technique is chromatography, where chromatography, their purpose is separate compound based on their distribution between a stationary phase and a mobile phase. This chromatography have three types, where the first type is thin layer chromatography, TLC, and the second type is high performance liquid chromatography, HPLC, and the third type is gas chromatography. The procedure of TLC is apply a small spot of the mixture on a TLC plate, develop the plate in a solvent, and visualize the separate components. And the procedure for HPLC, inject the sample into the HPLC system, where it passes through a column with stationary phase, and then detect and collect the separate component. The procedure for GC, inject the sample into the GC, where it vaporizes and travels through a column with a stationary phase, and then detect and record the separate component. The last chemical separation technique that I got is electrophoresis, where their purpose is to separate charged particles based on their size and charge. Their procedure is prepare an electrophoresis gel, where like agarose or polyacrylamide, and then load the sample into well in the gel, and then apply an electric field across the gel, and then smaller or more highly charged particles move faster through the gel. Stain the gel to visualize the separate components.
So for the sixth one is perform the measurement. The first one is calibration. Microplastic particles observed on the filter paper were identified by physical examination using a standardized color sorting system according to the characterization protocol provided by Hidalgo's ETAL. EDS analysis indicated the presence of heavy metals such as cadmium and aluminium on the MP surfaces, further highlighting their potential to adsorb other co-pollutants. The study identified different polymer types based on color of MPs through ATR-FTIR analysis, associating them with clothing materials like PE, PP, and PES, as well as degraded fishing groups around the coastal area. In addition, advanced spectroscopic techniques like micro-Raman and micro-FTIR offer significant advantages for characterizing unknown polymers and smaller MPs. For this one is validation, controls, and blanks. For validation, yes, it is valid. Controls, the shape of MPs can provide essential information for determining their origins and potential sources. Next, the collected MPs were categorized based on their observed colors, resulting in 7 distinct color classification. Blanks. Blank is used. Next, all filters, including blanks, were visually examined under a stereo microscope with no sample pretreatment, in consideration to minimize the loss of sample and the alteration of actual characteristics of MPs. Procedural blanks, n equals to 4, were used during sample processing to avoid MP contamination from external sources and handled in the same way as the samples. Analysis of the blank filters confirmed the absence of MP in these samples. The last one is replicate. The sampling was done at 4 different locations. First is faculty area, second is campus, the third one is research institute, and the last one is library.
last one is about result. This is analysis of MPs for 4 selected samples. The first one is blue microfiber with fractures. The second one is black microfiber forms crack or groove. And then this one is red microfiber with evidence of contamination by boron and silicon. And the last one is the transparent microfiber showing the presence of round-shaped biofouling. This is the color distribution of MPs in the different sampling site. Okay, this is the last one, which is ATR FTIR spectra of MP for 4 selected samples. The first one is blue MP, the second one is black MP, and then this one is transparent MP and red MP.